Welcome to this explainer. Today, we're taking a cinematic deep dive into the microscopic, highly electrified world of muscle biophysics. We're going to explore the incredibly delicate balance of ions and proteins that allows us to move, run, and even just stand still. We're looking at the very literal yin and yang of muscle physiology.
Imagine this, a tiny drop in your potassium levels, the same electrolyte you get from eating a simple banana, literally paralyzing a perfectly healthy muscle. Crazy, right? We're not just talking about feeling a little tired or fatigued, we're talking about total, flaccid paralysis. So how does that happen? Keep that in the back of your mind because we're going to solve this high-stakes physiological mystery together as we work our way through the evidence.
All right, let's kick things off with Section 1, the spark of movement.
So, before you even twitch a muscle, there's this incredible electrical balancing act going on. It's called the resting membrane potential. Think of every muscle cell in your body as a tightly wound spring, locked in at exactly negative 90 millivolts. It maintains this electrical baseline by being super picky about what it lets in and out, mostly sodium, potassium, chloride, and calcium. The cell is constantly pumping these ions back and forth just to maintain this precise negative charge, so it's primed and ready to fire in a split second.
And when a signal actually arrives from your brain, a chain reaction called excitation-contraction coupling kicks in. First, an action potential rockets down what we call T-tubules at a blistering 3 to 5 meters per second. Next, a voltage sensor called the DHP receptor detects that sudden electrical shift. It physically bumps into the ryanodine receptor, forcing it to open. And boom, the floodgates open. The sarcoplasmic reticulum unleashes a massive wave of calcium into the cell. Basically, your body just turned an electrical spark into a massive chemical flood.
Which brings us to Section 2, calcium, the contraction engine.
If that initial electrical signal is the ignition, calcium is absolutely the biological gas pedal. It latches onto a regulatory protein called troponin C. When it binds, it triggers a physical change, literally yanking another protein, tropomyosin, right out of the way. By moving that blocker, it exposes binding sites on the actin filament, letting the myosin heads grab on, pull, and generate the physical force of a muscle contraction. Without calcium moving that roadblock, your muscles just wouldn't be able to contract at all.
But all that power definitely comes at a cost. Researchers actually put it perfectly. Cells go to great lengths in order to contain, store, and profit from the signaling actions of calcium. It is incredibly potent stuff. If calcium was just left floating around in your cells, your muscles would be locked in endless spasms and rigor. It literally has to be strictly contained to stop cellular chaos.
And that leads perfectly into Section 3, magnesium, the relaxation master.
So how do we stop a contraction? Well, the cell relies on a continuous molecular tug-of-war. Calcium is the initiator, binding to site 2 of troponin C to start things off. But here comes magnesium, acting as the antagonist. It actually competes for that exact same spot. Just by being there, magnesium lowers the protein's affinity for calcium by almost 1.5 times, effectively loosening calcium's grip. On top of that, magnesium blocks those ryanodine receptors we talked about earlier, which slows down any new calcium from entering.
But just competing for binding sites isn't enough. The cell literally has to sweep the calcium away. Enter the SARCA pump. This heavy-lifting enzyme uses energy, specifically magnesium-bound ATP, to forcefully shove calcium back into storage. And get this, it's pumping against a massive 15,000-fold concentration gradient. It takes an unbelievable amount of cellular energy just to force a muscle to relax. Relaxation isn't passive. It's a highly active, highly demanding biophysical process.
What's really fascinating is how our bodies regulate this SARCA pump using 2 specific peptides. In the heart and slow-twitch muscles, you have phospholamban, or PLN, which basically hits the brakes on SARCA until adrenaline tells it to hurry up. But look at sarcolipin, or SLN, in skeletal muscle. It binds to SARCA and actually makes it uncouple. That means the pump burns up ATP energy without actually moving any calcium at all. Why would the cell purposely waste energy like that? To generate heat. It's a built-in biological mechanism to keep your body warm without shivering.
Moving right along to Section 4, the potassium balancing act. Remember that resting membrane potential we started with? Well, the ratio of potassium inside versus outside your cells is the master switch for it. If potassium outside the cell gets too high, a condition called hyperkalemia, the membrane depolarizes. It gets less negative, which actually forces the sodium channels to permanently shut, leaving you extremely weak. On the flip side, if potassium drops too low, hypokalemia, the cell hyperpolarizes. It gets so negatively charged that it needs a massive, almost impossible stimulus just to fire, again leading to profound weakness. Just a tiny shift outside the normal range can completely shut down your ability to move.
Let's see what happens in Section 5, when the system short circuits.
So what happens when these delicate ion channels completely fail? Well, take hyperkalemic periodic paralysis, or hyper-KPP. It's a genetic mutation where the sodium doors, the NaV 1.4 channels, literally get stuck open when potassium levels rise just a little bit, like after a really heavy workout or a big meal. Because the door won't shut, sodium just keeps pouring in, keeping the cell permanently depolarized. The muscle is trapped in flaccid paralysis until your body's homeostatic systems can eventually restore the balance.
And here is where we finally solve the mystery from earlier. Let's contrast hypo-KPP with hypo-PP, hypokalemic periodic paralysis. We just established that low potassium should normally make a cell hyperpolarized and really hard to fire. But in hypo-PP, a mutation creates a leaky gating pore in the channel. When potassium drops below 3.0, this leak causes a paradoxical depolarization. The cell just short circuits. The normal sodium channels are inactivated, and the person gets hit with sudden, terrifying paralysis. So, mystery solved. It's a mutant electrical leak triggered specifically by low potassium.
Now, paralysis is one extreme, but what about spontaneous cramping? That's where tetany comes in. This happens when the balance shifts and nerves become hyperexcitable, most commonly because of hypocalcemia, low ionized calcium in the blood. Without enough calcium stabilizing the outside of the nerve channels, they just start firing all on their own, causing involuntary spasms. This is actually why hyperventilating during a panic attack can make your hands cramp up. Changing your blood pH literally lowers your active calcium levels.
Okay, let's bring it back to reality with Section 6, the dietary electrolyte equation.
If you've ever had a cramp during a hard run, you've probably experienced the dehydration and electrolyte theory firsthand. As you sweat and lose water, the interstitial fluid space around your muscle cells literally, physically tightens up. That structural squeezing disrupts those membrane pumps we spent so much time talking about, creating a chaotic environment that triggers your nerves to misfire.
Your absolute first line of defense against all this is your daily diet, and we can map your exact requirements directly to the cellular roles we just learned. You need adequate sodium to generate that initial electrical spark. You need thousands of milligrams of potassium to maintain your resting membrane potential. Calcium is crucial to stabilize nerve channels and prevent tetany. And you need about 350 to 400 milligrams of magnesium to power those circuit pumps and act as the antagonist so your muscles can actually relax. But here is a really important evidence-based warning for you. More is not always better. If you consume highly concentrated electrolyte supplements, basically dumping massive amounts of sodium into your system without drinking enough water, you can induce hypernatremia. This throws your extracellular fluids totally out of whack and can paradoxically trigger the exact chaotic cramping and muscle interference you were trying to prevent. The biophysics demand precise balance, not just raw volume.
Which leaves us with one final thought. The next time you're pushing your body to the limit, whether you're at the gym, out for a run, or just dealing with daily physical stress, ask yourself, are you actively fueling the delicate electrical and biophysical balance of your cells, or are you just quenching your thirst? Understanding the true yin and yang of calcium, magnesium, and potassium gives you the power to really optimize the remarkable machinery inside your muscles. Thanks so much for joining me for this explainer, and I hope you never look at a glass of electrolyte water the same way again.